Hey guys, we're solving this biology paper today. This is October-November 2021, 9700, paper 1-2. Okay, so I'll be doing the whole 2021 series, by the way, very soon. There are a few changes, right? It's no longer 1 hour, it's 1 hour 15 minutes from 2022, and there have been slight changes to the infectious diseases and epidemiology chapter, right? Some diseases are no longer there. We have HIV, malaria, cholera. All right. So let's begin. Before that, I just want to talk about the threshold. This is paper 1-2, right? So A was at 27, B was at 24, C was at 20, D was at 16, and E was at 13. Okay. So just to be safe, you could assume that A star is looking at the difference 3 to 4 marks above, right? At 30 or 31. Or just to be safe, it's like 40 is the highest mark, right? So 40 minus 27 is 13, 6 marks above this. So A star was at 33 or 34. That's the safest bet, all right? The 90th percentile. Starting from 1. A scale bar on an electron micrograph is 2 centimeters long and represents an actual length of 1 micrometer. So we know how to do this. Magnification is equal to image length by actual length. So magnification is equal to image, right? 2 centimeters. 2 into 10 to the power minus 2 divided by the actual length, which is 1 into 10 power minus 6. Micro is 10 to the power minus 6. So 2 into 10 power minus 2 divided by 1 into 10 power minus 6. That gives us a value of 20k. 20,000 x 20,000 c. The eyepiece of a microscope is fitted with an eyepiece reticle, and a stage micrometer scale is placed on the microscope. Which statements about the stage micrometer scale are correct? The scale can be used to measure the actual length of cells directly. Not really. Not really. It can't. You need both. Okay. You need the help of the eyepiece reticle as well as the stage micrometer to measure the actual length of the cells. So 2, 1 is actually wrong. 1 is wrong, okay? This can't be right. So A and C are gone. The scale allows you to calibrate the eyepiece reticle, that is true. Less of the scale is visible as the objective lens changes from 10 to 40. Right. This is also true. I'll show you something. Wait up. Uh, we're talking about the stage micrometer, right? Stage micrometer at different magnifications, right? Let's see. Here we go, guys. I have a good figure for you. Oh, wait. Just messed up. It's kind of like this. I need a better diagram. Wait. Is this better? So as you can see, what's happening basically, as we zoom in from 10x to 20x to 40x, the stage micrometer, before we could see 80 divisions, right? Now at 20x we're seeing 40 divisions, and at 40x we're seeing 20. That's the stage micrometer. But what about the eyepiece reticle? We always see the same divisions of the eyepiece reticle. That never changes. We need both to find out the actual length. Okay, so 2 and 3 are correct. Less of the scale is visible as the objective lens changes from 10 to 40. If it was for, you know, for an eyepiece reticle, 3 would have been incorrect. But since it's for stage micrometer, This is correct. So 2 should actually be B. Four students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. mRNA passes through to the ribosome. Okay, so that's through the pores in the nuclear membrane. Okay, the pores in the nuclear membrane organizes microtubules to produce the spindle during cell division. Okay, so they're talking about the centrioles. Okay. 3. Synthesis of polypeptides. Ribosomes are responsible for the synthesis of polypeptides. Let's begin. So, this is basically the nuclear membrane. So, the nuclear membrane or the nuclear envelope is a double membrane structure. So, we're going to look for a double membrane structure, and that only matches with X, right? So, the X is actually 1. X is 1. Now, organizes microtubules to produce the spindle during cell division. We're talking about centrioles here, right? So, centrioles are actually non-membranous. Okay, they're non-membranous and cylindrical. Okay, so this should be 2. We have our answer. And ribosomes, this is centriole. Okay, and this is a ribosome. A ribosome is also non-membrane bound, but it's spherical in nature, right? It's spherical, so that's a W. 3 is W. What about V and Z though? Z, membrane bound sacs arranged as flattened stack. This is probably the Golgi body. It's in charge of packing. V, membranes which surround an enclosed inner cavity. It's probably a lysosome or a vesicle. Okay. Next, 4. Which cell structures are required for the formation of lysosomes and hydrolytic enzymes that they contain? Okay, mitochondria is required for energy. Rough endoplasmic reticulum, along with the help of ribosomes, is required for protein synthesis. And Golgi body is required for packing them into lysosomes, right? So B is the answer. All 3 are required. Which cell structures contain rRNA? Okay. Chloroplast and mitochondria contain rRNA. This is because, according to the endosymbiont theory, they were bacteria originally, so they have 70s ribosomes and rRNA. Now, the nucleus must also have rRNA. Okay, it's produced there. RNA is produced in the nucleus. However, smooth endoplasmic reticulum does not. Moving on. The single-celled organism, Thiomargarita namibiensis, measures 700 micrometers in diameter and is enclosed in a cell wall. The genetic material of namibiensis is located free in the cytoplasm, where it occurs as thousands of copies of circular DNA. Interesting. So this is probably prokaryotic. Most of the cell is taken up by a large vacuole which stores essential chemicals for metabolism. It must be a eukaryote because the diameter of 700 micrometer is too large for a prokaryote. This is clearly wrong. I told you it's a prokaryote. It must be a plant because it contains a vacuole. No way. A lot of other things contain vacuoles. It must be a plant because it is enclosed in a cell wall. That's wrong. Other organisms may have cell walls. It doesn't have to be a plant. Fungi have cell walls, for example. Okay, and even some prokaryotes do. It must be a prokaryote because its DNA is bacteria contains cell walls. By the way, okay. Fungi have chitin in the cell walls, and bacteria have mucopolysaccharides. It must be a prokaryote because its DNA is circular and located in the cytoplasm. Exactly what I was talking about, right? So 6 is D. That's right. Moving on to 7. A student carried out 4 tests on a sample of biological molecule. Iodine turned yellow. I mean, it is yellow in the first place. Okay, iodine is yellow in the first place. Yes, so iodine is yellow in the first place, right? So it doesn't really matter. So starch was not present because iodine did not turn blue black. Okay, so 4 is correct. Now the Benedict solution actually turned yellow, so reducing sugar or glucose was present. So, like, here's the thing. Um, if Benedict solution turns from blue to yellow, that tells us that a reducing sugar is present. Now, how are you gonna be sure that it's glucose? It can be any other reducing sugar. Okay, so I don't think 2 should not be correct because we aren't sure about glucose. It is a reducing sugar, but we are not sure that it's glucose. Okay. Moving on, biuret solution, it turned purple, so protein was present, and the emulsion turned cloudy. Okay, so fat was present. So 1, 3, and 4 are actually correct. 2 is technically wrong. Okay, which general formula is correct for monosaccharides such as fructose? Okay, so we have C6H12O6. Now we're talking about monosaccharides, right? So the ratio of monosaccharides will always be like it's always H2O. Okay, so it's like this: C H2O followed by N. This is for a monosaccharide. Okay, we have to conserve that ratio of water, the 2:1 ratio. Okay, that's the basis of a carbohydrate. Which statements can be used to describe the structure of cellulose? So it is a polymer made up of 1,4 beta 1,4 glycosidic bonds. That's true. It is a polysaccharide of hexose monomers. That is also true. It is unbranched. That is true. Made up of beta glucose. Perfect. Okay. 9A. 10. Complete hydrolysis of polysaccharides requires all the glycosidic bonds between the monomers to be broken. Okay. Enzyme X only breaks 1,4 glycosidic bonds. Which row shows how completely enzymics can hydrolyze molecules of glycogen and amylose? So here's the thing: both glycogen and okay, so glycogen contains both alpha 1,4 and alpha 1,6 glycosidic bonds since it is branched. So uh, all of it will be hydrolyzed more or less. No, all of it won't be hydrolyzed since it contains both alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds. Some of it will be hydrolyzed, not most. Some of it will be hydrolyzed. So it can't be this, it can't be these either. 10 should logically be A. Now what is amylose? Starch has two components, amylose and amylopectin. So amylose has only one form. Okay, alpha 1,4 glycosidic bonds. Amylopectin has alpha 1,4 as well as alpha 1,6. Do you understand? That's why for amylose, this enzyme can break down most of the bonds. Okay, so double plus. Okay, 11. Which molecules contain at least two double bonds? Okay, we need at least two double bonds. Saturated fatty acids. Saturated don't have double bonds. Okay, they're talking about the fatty acid part. Okay, so saturated does not have it. Which molecules contain at least two double bonds? Oh, at least two. Okay, so let's think about this. Which one's the best answer? So basically, saturated is out of the question. Okay, we're not gonna talk about saturated. So that's how we are gonna go to C. Hemoglobin and collagen only, right? Because saturated can't be it. Okay, the saturated fatty acid does not have a double bond. The saturated fatty acid is like this: COOH followed by CH2, CH2, CH2. Basically, there's only one double bond. There are not more than one double bonds. Okay. 12. The diagram shows an amino acid. Which group is changed to produce different amino acids? So we're looking at the R group. Okay. This is constant. This is constant. This is constant. This group, right? Basically, this will change. Valine, lysine, glycine. The CH3 group. Okay. You need to remember that. 13. Which is correct about the structure of proteins? Primary, determined this by the sequence of nucleotides. That's true. Occurs by hydrogen bonding between NH and CF. Okay, that's true. Tertiary, disulfide, ionic, hydrogen bonds, and hydrophobic interactions are involved. That is true. Always formed by association of two or more polypeptides. True. 13 looks like A. Why are the other wrongs? Do you want to know? Um, the number of amino acids. It's not the number, it's actually the sequence. Okay, which one comes after the other one? In a single polypeptide, it is either an alpha or beta 6. This is also wrong because both can be present at the same time. Okay, the others look okay. only globular proteins have this structure. Wrong. Fibrous do as well. Okay, fibrous proteins have that as well. Now D, the sequence of amino acids. This looks correct. A beta pleated sheet is the result of a folded polypeptide forming hydrogen bonds between adjacent strands. True. Reactions between side chains of amino acids gives it true. Always formed by two or more polypeptides and a metal ion. This is not always present. It's not compulsory. Okay, so D isn't the correct answer. 13A. 14. Which graph shows how possible changes in energy levels as a chemical reaction progresses with or without an enzyme? Okay, this is from chemistry. The enzyme basically reduces activation energy. So the dotted line should be below. Okay, so these are wrong. Sorry, with enzyme, that's the bold line, right? So with enzyme the activation is lower, activation energy is lower, so the bold line should be below the dotted line. All right, the bold line should be below the dotted line. And about A and C, which one is correct, right? So both the dotted line and bold line will end at the same point, same height. That's the important part. Okay, so A is wrong, C is correct. Okay, 15. The graph shows the relationship between the concentration of substrate and the rate of an enzyme catalyzed reaction, which shows how Km and Vmax for this enzyme would be affected if the same reaction was carried out in the presence of a competitive inhibitor. So what happens when you use a competitive inhibitor? Any clue, guys? Basically, the graph becomes like this. Okay, so it takes longer. It takes longer to reach Vmax. Do you understand? But Vmax remains the same. So Vmax remains the same. However, it takes longer to reach Vmax. So look at this is Vmax. This is half Vmax. Look at this. Compared to before, what happened to the Km? This was the old Km. Now look at the new Km. It is higher. It increases. Remember that. 15B. Very important MCQ. Okay. Important for paper 2 as well. 16. Liver cells contain vesicles that have proteins in the membranes which are specific for the transport of glucose. When these cells take up glucose, the vesicles fuse with the cell surface membrane. How does the uptake of glucose occur? So, fusing with the membrane like this, when vesicles fuse, that's called endocytosis. Okay, fusing is called endocytosis. But look at this. Liver cells contain vesicles that have proteins in their membranes which are specific for transport of glucose. Now, basically, they are not talking about the process where the vesicles fuse. Okay, you're going to learn about this in A2. Basically, there are some vesicles containing some transport proteins. What happens is when we require glucose, the vesicles mainly fuse with the membrane. Okay, after fusion, we incorporate the transfer proteins into the membrane. Now glucose can enter the cell through these transfer proteins down the concentration gradient passively without using energy. So that process is called facilitated diffusion. 16D. 17. A student set up an experiment to investigate diffusion. A block of agar 1x 1 centimeter was stained uniformly with a water-soluble blue dye. The block of agar was put into a test tube containing 10 centimeter cube of distilled water. The intensity of the blue color of the water after 5 minutes was measured. Okay, so the blue color will basically come out of the block of agar and come into the distilled water, right? 4 experiments A, B, C, D were then carried out using different numbers of agar blocks, different sizes of agar blocks, and different temperatures. All of the variables were standardized. Which experiment would give a lighter blue color in the water after 5 minutes compared to the first experiment? Okay, so we want a lighter blue color, not a deeper one. Lighter blue means less diffusion took place. Okay, less diffusion took place. So I'll teach you which one would have the darkest color. So if you wanted the darkest color, we need the highest temperature, right? And the greatest number of blocks. Okay, so yeah, that would be A. That would give the darkest color. Now let's go to mm, the lightest color. For 17, we want the lightest color. So we're not going to look at the higher temperatures. We're going to look at 20 degrees. Now it's between B and D. Interesting. So 8 and 2 were comparable, but 2 and 1. To do this, we actually need to look at the surface area to volume ratio. Okay, look at the surface area to volume ratio. For D, what is the surface area to volume ratio? The volume is basically 2 into 1 into 1, which is 2. This is D. And the area is, it's basically like this. 1 is 2. The others are 1. So it's basically 2. One side is 2 centimeters basically, right? One side is 2 centimeters. So one side is 2. The others are 1. So it's 2 into 1, right? Into 2. Plus 1 into 1 into 2. Plus 1 into 1 into 2. How much is that? Basically, it's 8 centimeters square. Okay, so the area is 8, and the surface area to volume ratio is 8 is to 2, which is 4. Okay, so the greater the surface area to volume ratio, the greater the diffusion. Now look at B. For B, the volume is zero point 0.5 cube, 0.5 cube, so that's 0.125. Okay, what about the area? The area is zero point 0.5 into 0.5, right? Basically into 6. All the sides are the same. 0.5 into 0.5 into 6. That's actually 1.5. Okay. So there are 2 blocks though, right? In total, there are 2 blocks. So if we think about the whole thing in total, the area will be multiplied by 2 and this will be multiplied by 2. Okay. So the total surface area is 3 and the total volume is 0.25. So 3 by 0.25. How much is that? 3 by 0.25 is 12. Okay. So that's it for B. Now, the interesting part is in 17, we need to compare this to our initial experiment. Okay, we need to compare this to our initial experiment. In our initial experiment, we had this block, right? It had a volume of 1, right? And a surface area of 1 into 1 into 6. So that's 6. So the surface area to volume ratio was 6 by 1 or just 6. Now, out of these 4 other experiments ABCD, which were carried out using different numbers of blocks, different sizes of blocks, and different temperatures, everything else was standardized. Which experiment would give a lighter blue color in the water after 5 minutes compared to the first experiment? So yeah, we aren't taking A and C. I clarified because the temperature is higher, we'd get a darker color. So we were confused about B and D, right? We're confused about B and D. So in D, the surface area to volume ratio is 8 by 2, which is 4. And in that's in D. And in B, it's at 12. So the greater the surface area to volume ratio, the more likely that the color will spread, right? That's the logic. So how do you approach this question actually? Rather than looking at the rate of diffusion, okay? You need to understand that the key point here is a block of agar was stained uniformly. You understand? We are not looking at the rate of diffusion, okay? Comparatively. Basically, rather than the surface area to volume ratio, according to the examiner's report, I'm going to link it down in the description. You can look at the report for paper 1 2. The volume of the dye matters really. So in the original box, in the original block, sorry, of agar, we had 1 centimeter cube of agar. Look at these, right? Here we have 0.125. This is also 0.125. Okay, what about this? What was this? 1 into 1 into zero point 0.5. That's zero point 0.5 centimeter cube. And for D, we have 2 into 1 into 1, which is 2 centimeter cube. That's the logic. So I already told you, in the two options at 30 degrees, why are they not correct? Okay, so here's the thing. Why are the number of blocks given? The values I wrote on the left hand side, right, these are for one block only. You need to calculate it for all blocks. So if you multiply this by 8 into 0.125, that gives you a value of 1. Okay, then 0.125 into 2, that gives you a value of 0.25. Okay, next 0.5 into 2 gives you a value of 1, and 2 into 1 gives you a value of 1. Do you get it? So the logic is, um, it can't be A, right, because it has the same volume and greater temperature, so it will diffuse out faster, it will be darker. It can't be C because it has a greater temperature and the volume is also greater. It's 2 compared to 1, right? It's 2 compared to 1, clearly. If you multiply 2 with zero point 0.5, you're gonna get, sorry, if you multiply 2 with zero point 0.5, you're gonna get 1. That's also 1. Okay, so A and C are actually both 1. And the temperature is greater. The same volume and temperature is greater, so it will diffuse out faster. Now look at B and D. Out of these two, which one is the correct answer? We should go for B. I'll tell you the logic. Look at this. Why should we go for B? Because in B and in D, it's actually 2 into 1. Right? It's 2 into 1, which gives you a value of 2 in D, right? That's actually, wait, let me do the math again. Wait up. Yeah, we don't need to think about the surface area to volume ratio here. Okay, we didn't need to do that. For 17, B will be the answer. Okay, B is the answer because hear me out. Look at this. 0.5 cube is 0.125, right? It's 0.125. And there are two blocks, right? So multiplied by 2, that gives you a value of 0.25. For B, it's actually 0.125 into 2, which is 0.25. So this is much less than the initial volume of 1 cm cube. Okay, and the temperature is also the same, so it will diffuse out at the least speed. What about the first one? It's 0.5 cube into 8, which gives a value of 1 cm cube and greater temperature, so it will diffuse out faster. For C, it's zero point 0.5 into 1 into 1 into 2, which gives you a value of 1 cm cube as well, greater temperature. For D, it's 2 into 1, so that's actually 2 centimeter cube. Okay, same temperature but 2 cm cubes, so it will become darker clearly. Okay, moving on to 18. Which description is correct? A centromere holds two chromatids together until the end of prophase and attaches to the microtubules of the spindle. No, not really, because it holds them together till the end of metaphase, till anaphase begins. A chromatid is one of the two identical parts of a chromosome and is made up of proteins and two molecules of DNA. That's wrong. It's made up of nucleotides and one molecule of DNA, okay? One chromatid is one molecule of DNA. Next, a chromosome is a structure with two identical parts made of DNA and proteins found in the nucleus of a prokaryotic cell. Prokaryotic cells do not have nuclei. Come on. A telomere is a sequence of DNA sequences such as GGG, TAA repeated many times and found at the ends of each chromatid. This looks good. Prevents aging, right? 19. During metaphase, a scientist stains the chromosomes of a diploid animal cell with fluorescent dye to allow the telomeres to be observed. This cell has 26 chromosomes. It has 26 chromosomes. Things like these. This is one of the chromosomes, okay? So each chromosome has two chromatids, all right? Right, so each chromosome is made up of two chromatids, okay? Now, each chromosome contains, like each chromatid contains 2 telomeres at each end, right? So one chromosome contains 4 telomeres. So if you multiply 4 with the number of chromosomes, 26 into 4, that will give you a value of 104, and that should be your answer. 19 should be D. Moving on to 20, the photomicrograph shows cells in various stages of the cell cycles. Which cells contain twice as many DNA molecules as a cell from the same organism that has just finished a complete mitotic cell cycle ending with cytokinesis? So we are seeing, you know, this is metaphase, prophase, anaphase, and telophase. So here cytokinesis has not occurred. All of them contain double. As long as you haven't divided the cytoplasm, you will get double the number of chromosomes in each, double the number of DNA molecules, right? Twice in each cell. 21. Uncontrolled cell division can result in the formation of a tumor. Which part of the cell cycle would take less time during the formation of a tumor? Basically, when is DNA doubled? It's doubled in the S phase in interphase, right? So there is an issue in the S phase which leads to uncontrolled mitosis, tumor formation. So 21 is B. 22. A piece of a DNA molecule contains 84 base pairs. The table shows the number of adenine and cytosine bases in one or both of the DNA strands in this piece of DNA molecule. So, okay, how many guanine bases are present in this piece of DNA molecule? Interesting. It contains 84 DNA base pairs. 84 base pairs, all right. So we have two strands of DNA. In strand 1, we have adenine 28, right? So in strand 2, okay, I'm going to do this. We have thymine as well, and we also have guanine. So in strand 2, remember that adenine binds to thymine and guanine binds to cytosine. So in strand 2, we're going to have 28 thymine, and in strand 1, we're going to have 23 thymine. So it should match, okay? Now, for cytosine, we have 15 in strand 1. So in strand 2, we should have 15 guanine. Now the logic is a piece of DNA molecule contains 84 base pairs. Okay, so this should be 23, right? All of these need to add up to form 84. So do this. 28 plus 23 plus 15 plus guanine is equal to 84. So 84 minus 28 minus 23 minus 15. Guanine is a value of 18 over here, 18, right? And it should also be 18 over here. It should match. So guanine in total, right? In total, 18 plus 15, that is equal to 33. Okay, so 22 should be B. See if you guys understand what I did. Okay, I first matched adenine and thymine complementary base pairing on two different strands. Then what did I do? Then I matched cytosine with strand 1 and 2, and then I subtracted from 84 to get the number of guanine, and then I just added them up. Okay, 23. The mRNA sequences of the three stop codons are shown. Which mutation in the template strand of a DNA sequence that codes for a polypeptide would cause translation to stop prematurely? Okay, so we're looking at the DNA strand, right? ATT is changed to ATC. So this is the DNA strand. Look at the mRNA then. This should be, in mRNA we have, instead of adenine, instead of thymine, we have uracil, right? So adenine should be base paired with uracil followed by adenine, then guanine, UAC. This looks good. This should be UGU. Okay. Then what about this? This should be A binds with uracil, thymine binds with adenine, and this thymine binds with adenine. So yeah, here's the issue. This turns to UAA, right, in 23. So this is a stop codon. C should be the answer. Which features of component cells are essential to the function? They're connected to safety elements by plasmodesmata. This is essential, right? Because the safety, right, and the this is the safety element, and this is the companion cell. We need to transfer, after loading, the sucrose needs to go into the safety element passively, so this is required. They have thicker cell walls than a safety element. What role does this have? This is wrong. 2 is actually wrong. Okay, they contain a nucleus and mitochondria, right? So they are actively pumping in, loading sucrose, right? We need energy for that. Of course we need that. Okay, so 1 and 3 are correct. 24 is B. 25. Which statements about water movement in plants are correct? Water can pass through cellulose cell walls. Okay, that's true. Water cannot pass through lignified cell walls. Remember, there are two pathways, the apoplastic and symplastic. So yeah, water can pass through cellulose cell wall. That's true. Water cannot pass through lignified cell walls. That is true. Water can't pass through the lignified cell walls of, xylem. So 2 is also true since water cannot pass. Okay. Now, what about 3? Water can pass through cells that contain suberin. No, remember the Casparian strip? Water cannot pass through that. Okay. So 25B is correct. It cannot pass through suberin. 26. The diameter of a tree trunk usually decreases slightly during the day. Which changes in environmental factors during the day could cause the diameter to decrease even more? Okay, so why does the diameter actually decrease? It's due to the transpiration stream, capillarity, because water is pulled up, right? It causes a strain on the diameter and it decreases. So we want to look at things that actually increases transpiration. So increased temperature does increase transpiration. Increased wind speed increases transpiration, but increased humidity actually decreases the steepness of the water gradient. So increased humidity is wrong. Temperature and wind speed are fine. So 26 should be B. 27. Which sequence of events could lead to mass flow in a phloem sieve tube? Okay, let's see. Sucrose moves into a sieve tube element. Oh, it's basically the sequence of events, right? This happens, yeah, not at the beginning. Let's see. Protons and sucrose molecules move into companion cell through a co-transfer protein. Okay, this happens in the beginning. Hydrostatic pressure is produced. Water potential of sieve element decreases. Protons are actively pumped out. Water moves down a water proton. Okay, so initially what happens is this is the sieve tube element, and this is the companion cell, and these are the leaf cells. Okay, so initially we pump out H plus into the cell wall. So this one will go first. We pump protons into the cell wall actively. Then the protons along with sucrose uh, move into the companion cell through the co-transporters. So 2 is actually 2. Then after that moves in, okay, after we have 5, 5 comes first, then 2. 5 comes first, then 2. Then sucrose moves into the sieve tube element by just passive diffusion. That's step 3. Okay, now what? As sucrose entered this, water potential will decrease, right? Water potential of the sieve tube element decreases. 4 is 4. Now what? Water will move down the concentration gradient into the sieve tube element, and then a very high hydrostatic pressure is produced. That's the logic. Okay, remember this sequence, really important. 28. What will happen as a result of the blood pressure in the right ventricle becoming higher than the blood pressure in the right atrium? Okay, so when blood enters the right ventricle from the right atrium and pressure increases, the valve between the two regions, right? Okay, yeah. So for 28, I was saying that when the pressure in the right ventricle exceeds the pressure in the right atrium, basically due to this increased pressure, the valves between them will close. Okay, so the valves between the right side of the heart is the tricuspid valve. Okay, so that's the right atrioventricular valve, so that will close, okay, due to the increased pressure. So we can look at the four chambers. It's like this is the right atrium, right ventricle. Blood actually rushes in from the right atrium to the right ventricle. When the pressure here exceeds the pressure here, this valve closes. That's the right atrioventricular valve. Okay, 29. The graph shows pressure changes in different parts of the heart during a mammalian cardiac cycle. W, X, Y, and Z indicate when a valve opens or closes. So let's see. In 29, we have four options. So let's look at this. I'm going to teach you what actually happens. So this bold line, do you see this? This is actually the pressure in the ventricle, right? What about this dotted line? This is our atrium, and this is basically the aorta. So look at this. So at W, what actually happens in W is the atrioventricular valve closes. Okay, when the pressure in the ventricle exceeds the atrium, I just told you in the previous question, the atrioventricular valve closes. Okay, and right after that, what's followed? What will follow after this is when pressure increases here, the semilunar valve, right, from the ventricles to the lungs, maybe, or to the aorta, those semilunar valves will actually open. Okay, the semilunar valve opens at X. The semilunar valve opens. Right after that, what happens at Y? When the pressure slowly decreases, the semilunar valve will close, and then afterwards, again, when the pressure in the atrium exceeds that of the ventricle, then the atrioventricular valve opens again. Okay, so which matches with this? It should be option B. You should have this memorized. Okay, for 30, which statements are correct? Compared with blood, tissue fluid has less protein and no RBC. This looks okay. This is fine. Lymph may contain lipids, carbon dioxide, and phagocytes. That's true. Tissue fluid contains glucose, amino acids, urea, and carbon dioxide. That's fine. That's fine. Those can actually pass through the capillaries into the tissue fluid. So 30 is actually A. 31. Which statement about the formation of hemoglobinic acid are correct? It can only occur with the dissociation of oxygen with hemoglobin. That's true. Initially, we have HbO8. Then, when we get acidic conditions due to, H2 combining with CO2 to form H2CO3, and then H2CO3 breaks down to form H plus and the bicarbonate anion. Okay, so when this happens, um, basically H plus has higher affinity towards hemoglobin. Okay, so we're gonna get hemoglobinic acid, and the oxygen is released in the process of doing so. So it occurs after dissociation of HbO8, right? It first breaks down to form hemoglobin and oxygen, and then hemoglobin combines with H plus. Okay, so one is correct. It removes excess hydrogen ions, preventing blood, right? It acts as a buffer. It has a buffering action. It is linked to the action of carbonic anhydrase. Yes, carbonic anhydrase actually catalyzes this reaction that I was telling you guys about at the beginning. Okay, so 31 is A, all are correct. 32. What explains how the maximum volume of oxygen is taken up as blood? Sorry, how the maximum volume of oxygen is taken up as blood through the capillaries of the lungs. Okay, as blood passes through the capillaries of the lungs. Each hemoglobin molecule can temporarily bind to 4 oxygen atoms. This doesn't look like the best answer. The dissociation of carbon dioxide from carboxyhemoglobin allows more hemoglobin to be available for binding oxygen. We don't have much carboxyhemoglobin in the first place. Okay, the binding of the first oxygen molecule to hemoglobin decreases the affinity. That's wrong. It actually increases the affinity. Oxyhemoglobin formation increases the ability of, yes, that is the sigmoid shape. Basically, after one hemoglobin binds to one oxygen molecule, it becomes easier for the second molecule to bind, and so on. Now, this actually helps in the pickup of oxygen. Remember that, really important. 33. The image shown is a photomicrograph of a transverse section of part of the gaseous exchange system. So I'm going to teach you guys. Do you see this? This is the cartilage. How am I figuring this out? Look at the lacuna. These are the lacuna of the cartilage. This is probably the C-shaped cartilage in the trachea. Okay. What type of epithelium does the lungs have? Trachea initially and the bronchus. Pseudostratified ciliated epithelium. Okay. Why is it called pseudostratified? Even though it's a single layer of cells, it looks like multiple layers. Why is it ciliated? Look, you have the cilia for the sweeping action. And here you can see each cell. Do you see them? These are the cells basically, okay? These are your cells. We have goblet cells, then they secrete mucus and stuff, alright? They keep it lubricated. These are the cells. That's the lining. This part is the lining, alright? Now, just below the lining, we have the basement membrane. And after the basement membrane, look at this. Do you see this? We have the elastic fiber. We have some blood capillaries. Okay. So, what describes the image? A thin inner layer of ciliated epithelium on top of a layer containing cartilage supported by elastic fibers. A very thin epithelial lining with walls containing elastic fibers surrounded by many blood vessels, an inner layer of ciliated epithelium and goblet cells. Okay, this is what I was talking about. Not only is this ciliated epithelium, it also contains goblet cells. That's why A and B were wrong. We need to talk about goblet cells on top of elastic fibers supported by an outer layer containing cartilage. So this is in the outer layer, and this ciliated epithelium with goblet cells, right? This is actually the inner layer, all right, and supported by an outer layer consisting of cartilage. What's wrong with D? Inner layer of ciliated epithelium and goblet cells on top of loose tissue and mucous glands. Continuous ring of cartilage. No, there's multiple things wrong. There is loose tissue, but there are also elastic fibers, okay? But there's no continuous ring of cartilage. It's C-shaped or incomplete, okay? 33C is the best answer. Moving on to 34, which shows the tissues that are present in the wall of the trachea and the wall of the bronchus. Okay, so remember that we don't have squamous epithelium in trachea and bronchus. Remember that. That's present in alveoli. So there is only one answer which fits the criteria. We do have smooth muscle and goblet cells, so 34 is C. 35. When a person suffers an asthma attack, the tubes of the gas exchange system narrow and extra mucus is produced. So I don't think we have this in our syllabus, but I'll still teach you what happens during an asthma attack. Basically, I have asthma, so I know how it feels. The activity of the ciliated epithelium increases due to the inflammation. Okay, due to the inflammation, that is true. The sweeping action increases. Endocytosis, no, exocytosis increases. So 2 is wrong. Okay, 2 is wrong. Smooth muscles become more active. That's true. C. Okay, 36 is not in our syllabus anymore. Tar smoking, it's been excluded. Okay, 37. Which control measures would reduce the transmission of tuberculosis? This is true. Boiling water before drinking it. It's not transmitted by the fecal oral routes, so no. Okay. Antibiotics, it is a bacterium, so yes. Vaccination, yes. We take the BCG vaccine for TB at birth, right? Okay, so 37 is in fact D. 38. Comorbidity is where an individual has two or more diseases or medical conditions at the same time. We saw a question like this in February, March 2022, variant 22. Some medical conditions are particularly likely to result in comorbidity. In one example, initial infection with one pathogen can increase the risk of developing a second disease by a factor of 15 or more, as we see in HIV and TB. Which description could fit this example? HIV infection followed by the development of TB. This is totally perfect. Okay, 38C. 39. Which conditions are infectious and result in a high white blood cell count? Okay, let's try to understand. This isn't in our syllabus anymore as well because myasthenia gravis, it's an autoimmune disease. It has also been removed from your syllabus. Okay. So basically, you need to understand that leukemia is a cancer of WBC. So we actually reduce the number of WBCs. Okay, and myasthenia gravis is an autoimmune disease. It's not an infection. Only TB is an infection from here. 40. Which cell type causes the secondary immune response to be much faster than the primary immune response? This is mainly due to memory cells, right? They keep a memory of what happened in the past. Okay, so that's it, guys. If you have other questions, please comment down below. And if you like the content, please like the video and subscribe to the channel. It would help me a lot. I'm gonna link paper 1 up here, and I'm gonna link ON 2021 variant 1 up here, and I'm gonna link ON 2021 variant 1 3 down here when I solve that. And remember to hit the subscribe button. Okay, see you guys.